Department of Pharmaceutical Technology at LG Institute of Pharmacy, Ahmedabad. In this video, I will talk about the microbial staining and its importance, different types of staining techniques and specifically simple staining technique. This video lecture is part of the online education system implemented by the LG Group of Institutes, Ahmedabad for the upcoming academic session, and that is to avoid the physical classes for the safety of our students due to current COVID-19 pandemic situation. So let's see about the microbial staining. We all know that microorganisms play an important role in our lives. They are harmful, but some of them are also beneficial, so their study is important. But because of their micro size, we cannot see them with the naked eye. So their study required the use of microscopes. But most of the microorganisms are colorless and transparent. Even with the help of microscopes, these are very difficult to be observed. So if the contrast is there between the microorganisms and the background field, they can be easily visualized. And that is the role of microbial staining. This is the process in which the contrast is created between the microorganism and the background field of the microscope by coloring the microbial specimen with staining reagent. So that enables the better visualization of the microorganism. And we can study the shape, size and arrangement of the microorganisms. We can even differentiate the type of microorganisms. For example, using gram staining technique, we can differentiate gram-positive and gram-negative bacteria. We can identify the internal and external structure of the microorganisms. Even we can differentiate the living and dead cells using viable stains. We can study the distribution and chemical nature of the cellular components. So whichever is the purpose of the microbial staining, that depends on the type of staining reagent that we use and the type of staining technique that we implement. So stains or sometimes dyes, these are the organic compounds which bind to the cell or cellular components and impart the color to the microorganisms. The staining reagents in the structure contain the basic benzene ring structure along with the functional group that is responsible for the coloring, that is called chromophore, and that is attached with the oxochrome, that is the salt form, and that gives the ionic property to the chromophore. So oxochrome helps to bind the stain with the microbial cell surface or cellular components, and chromophore impart the color upon binding with the microorganisms. So based on the charge that is carried by the chromophore, stains can be classified in three categories. One that is called basic stain or cationic stain. In this stain, the chromophore carries positive charge, and the variety of the stains are available in market. For example, methylene blue in its chloride salt form, crystal violet, malachite green, basic pushing, saffronin, etc. The second type of stain that is called acidic stain or anionic stain, and in this stain the chromophore carries a negative charge. So the dyes like negrosine, Congo red, Indian ink, acetylphosgene, eosine, etc. can be used as a negative stain. The third type that is called neutral stain, because the chromophores in this stain carries both positive and negative charge. So the example like eosinated methylene blue or ginsa stain or white stain, etc. can be used as neutral stain. Stains are pure than the dyes, and for the biological purpose, certified stain should be used. Now let's see about the types of different microbial staining techniques. So basically there are two techniques, simple microbial staining technique and the another that is differential microbial staining technique. Now the basic difference between these two techniques is that the simple staining technique involves the use of only one staining reagent, whereas the differential staining technique involves the two or more than two different staining reagents. Simple technique generally used to identify the basic morphological characteristics of the microorganisms, whereas the differential staining technique is used to differentiate the organisms and to study the spatial structure of the microorganisms like flagella or spores or spirochetes type of organisms can be studied by this technique. Now if we talk about the simple staining technique, so depending on the type of stain that we use, it can be called as a simple positive staining technique or negative staining technique. Simple positive staining technique is also called direct staining technique or monochrome staining, as one coloring agent is involved. The negative staining is also called as relief staining technique. So simple positive or monochrome staining technique involves the use of basic dye or basic stain, whereas the negative staining technique involves the acidic type of stain. Differential staining technique, there are two popular techniques in this category. One that is gram staining technique that differentiate the organisms in gram positive and gram negative type of organisms. And the another one that is acid fast staining technique that differentiate the organisms as acid fast and non-acid fast. And this technique is specifically used to identify the mycobacterium type of organisms. Let's see about the simple positive staining technique. The principle behind the staining of the organisms in this technique is that the microorganism in neutral medium attains the negative charge, and that is maybe because of the anionic phosphate group present in chichoic acid or the lipopolysaccharide, and this negative charge is being attracted towards the positively charged chromophore of the basic stain. This technique involves the basic stain. So that is how the interaction of the basic stain with the negatively charged microbial cell surface or cellular components occurs, and that results into the coloring of the organism. So that's why this technique is called direct staining. So here in this diagram you can see the stains are usually available in their salt form. So upon ionization in the aqueous or alcoholic solution, this stain attain the positive charge. This is the chromophore functional group that is responsible for the color, and this positive charge is being attracted toward the negative charge, and because of this ion exchange reaction, binding of the chromophore with the cell surface or cellular component occurs, and that results into the coloration of the organisms. Let's see the procedure of simple positive staining. This involves 5 basic steps. One that is smear preparation, second that is heat fixation, third that is reaction with the staining reagent, fourth that is washout of the excess reagent, and the fifth step that is drying of the specimen. In this pictorial presentation or process, you can see that the first step that is smear preparation. Here you need the clean glass slide. You can mark with the marker the bottom part of the slide, and you need nichrome wire glue, that is to transfer the culture from the source to the slide. So if it is in a liquid consistency, you can transfer 1 loopful of culture that is microorganisms over the clean glass slide. If the culture is in solid consistency, you have to mix with the 1 drop of water over the glass slide, and then spread this culture in thin layer and allow it to air dry. This is called the smear preparation, that is spreading the microorganisms in thin layers so that we can see the individual organisms without any overlapping. The next step that is heat fixation. So with the help of slide holder, you can move the slide with smear facing up through the flame of the burner 3 to 4 times quickly. So that results into the heat fixation, that is the fixation of the smear to the glass slide because of the heat treatment. This heat denatures the protein of the microorganism and kill them, and that helps to bind the organisms to the slide. If the smear is not heat fixed to the glass slide, then it will be washed away with staining reagent in the next step. So that is important step. So now the next step, that is the reaction with the staining reagent. So you have to flood the smear with the staining reagent and allow the appropriate reaction time. That depends on the type of staining reagent. For example, if we use crystal violet, then we have to allow the reaction for 1 minute. If we use saffronin, then we have to wait for 3 to 4 minutes for the reaction that to happen. So after that, we have to drain the excess of the staining reagent and then wash out the excess reagent by keeping the slide in standing position under the running tap water or either from the bottle. So that is slow running water, and you have to drain the stay until you find the drain is clear of any coloring reagent. Then dry, whether you air dry or use blotting paper to dry the specimen. Now the slide is ready for the observation. You can observe under the 10x, 40x, and if the high resolution is required, then oil immersion lens can be used. But take care that the oil that is used should match with the refractive index of the glass, that is of the cover slip and glass slide. So what we may observe when you use this technique, that all the microorganisms get colored against the colorless background. Now the color depends on the staining reagent that we use. In case of methylene blue, then we can see all the organisms are colored with the blue color. If we use saffronin, then we can observe the pinkish red color of the microorganisms. So in this specimen, for example, the sample is of bacteria, then we can see that the shape and arrangement and size of the organisms. For example, in the diagram, we can see that the shape of the organism is round, and that is called cocci. So here in this diagram, we can see the arrangement of the organisms, the bacteria, the cocci in chain form that represent the streptococci, the cocci in bunch form that represent the staphylococci type of species. In this specimen, we can see the rod-shaped bacteria, that is called bacilli. So the basic information provided by this technique can be useful for the further confirmatory test or the identification test. But some of the microorganisms are difficult to be stained by the staining reagent, and in that case, this technique is not useful. So we can go for the another technique, that is the simple staining technique of negative staining type, and this technique involves the use of acidic or negatively charged staining, and the principle behind the negative staining is that the bacterial cell surface or that possesses the negative charge and that repel the negatively charged chromophore of the acidic stain. So that results into the repulsion of the stain from the microorganism, and rather the stain gather around the cells, and that results into the staining of the background instead of staining the organisms. So we can observe the colorless microorganisms against the colored background. So here I have taken the example of acidic stain, that is negro stain, and that is the water soluble version, that is also known as acid black too. So upon ionization, this sulfate group attains the negative charge, and that is repelled by the negatively charged cell surface or cellular components. Let's see about the procedure of negative staining. The procedure is simple. What you have to do, you have to take a clean glass slide, and the end of the clean glass slide, put a drop of acidic stain. I have shown the pictorial diagram of the procedure, and to this drop of acidic stain, mix the loopful culture of the microorganisms and mix it, and then with the help of another glass slide, hold it at a particular angle, and then spread this drop across the glass slide in thin layer and allow the air drying of this mixture of the organisms with the acidic stain, and then observe under the microscope. So if we compare the procedure of negative staining with simple positive staining, we can see that there is no heat fixation involved in this process and no washout of the excess reagent required. So when heat fixation is not involved, it is advantageous, and it is at the same time it is also disadvantageous. So when heat fixation is not there, the microorganisms are not being killed. So we can observe the shape of the microorganism without the distortion. That is the case with the heat treatment. But when heat fixation is not there, the microorganisms are alive, and that's why this technique is not suitable for studying the pathogens. Uh, so what we may observe when we use this technique, we can observe that the colorless organisms against the colored background. The background color depends on the staining reagent that is used. In this case, it is a nigrosin. So in diagram, we can see the shape of the organisms, the bacilli or foci type of organisms are present if the sample is from bacteria. So this is all about the simple microbial staining techniques. In my next video, we will see about the differential staining technique, that is lamp staining and acid fast staining. Hope you find this video interesting and useful. You can see this video and other videos related to the pharma topics on our YouTube channel, that is Pharma Ignite. So do visit and subscribe. Till then, thank you.